Good afternoon, everyone. So once again here, before you guys to share once again what Comvita do for trying to fight against the Varroa mite. Just wanted to let you know that at Comvita, our purpose is clear. Working in harmony with bees and nature in New Zealand to heal and protect the world. It is well known that populations are under threat globally. Loss of habitat due to the urbanization, climate change, industrial agriculture and monoculture, parasites, pathogens, and pesticides have all contributed to a heavy decline in the world's number of pollinators. It has now got to the stage where bee populations are in crisis. They depend on human intervention for their survival. To deliver on our aspirations, we have established a new set of parameters by which we hold ourselves to account and measure success. This is our Harmony Plan that values the association with our bees to commitment to develop, implement, and promote bee welfare results aligned with international best practices. The loyalty to our operating principles is demonstrated by our actions, especially with the choice we make. Many beekeepers' tasks can perform in different ways. Best practice comes from choosing the best option or developing new ways of working that align to our operating principles and deliver great bee welfare outcomes. These points are the agreed operational standards to our achieving the best practice at Comvita. First, we ensure our teams have adequate training, a safe work environment, and are well supported so they can focus on being good beekeepers. Second, we plan our activities and design our tasks such that our interaction causes minimal adverse response from the bees to us, the beekeepers, and activities, and achieves the 5 freedom outcomes. Our choice of actions described above will create the least disruption to the colony while maintaining optimal health outcome and colony strength. To control efficiently Varroa mites is very difficult for us too. And it's becoming very problematic because it's disrupting our hive management plans, but we have fight them in accordance with the Bee Welfare Code. That's why we come here to tell you that the fight against Varroa is also a high priority for us, and we are not isolated from the problem that afflicts beekeeping in New Zealand and around the world. The question I would like to leave on the table is, knowing that Varroa is disrupting our entire hive management plan to keep our beehives alive, what should all of us do together? Thank you.
Kia ora koutou katoa. My name is Hayley Pragert and I'm a Senior Advisor for the Ministry for Primary Industries, Biosecurity New Zealand. I know today is a Q&A session, but I'm really interested in hearing what you guys have to say about Varroa. When you open your hive, what are the big issues that you have? What are the issues that you face in managing this problematic mite? Biosecurity New Zealand conducts the colony loss survey every year in order to track issues that beekeepers face and track why beekeepers are losing hives. And hopefully you got along to Pike's talk this morning and learnt a lot about it, but from what we know about the colony loss survey, that in the past 7 years that we've been doing this survey, that Varroa has become an ever-increasing problem for beekeepers, and in 2021 and in 2022, it was the most significant pathogen for beekeepers. And so, after 23 years, I think a key question that we all want answered is, why is it a problem? So I guess today what we're really interested in finding out is that, and hearing your thoughts. And I guess that's really,
Kia ora. I'm Michelle Taylor. I'm a scientist at Plant and Food Research and have been working on Varroa for probably a large portion of my career. And my goal is to eradicate Varroa so I can retire once it's done. So I'm kind of keen for that to occur. So, pretty much this is where we've been, and we've possibly still got a long way to go. But I just wanted to show this so that we can understand and remember where we have been. So we have started with Varroa and its biology and teaching the industry how to manage it. And as an industry, you've done an incredible job of managing this pest for 23 years. Well done. Super proud that we didn't lose major colonies. We didn't have major colonies lost like we saw overseas. We didn't have areas where you'd have 50 to 90% colony loss, so well done. What we did look at is the symptoms, and we explored what it looks like within our colonies, within New Zealand colonies. We looked at what, you can see the little picture up there with deformed wing virus. We saw what effect that had on the industry when that came in 2017, somewhere in that sort of range. We developed a Varroa sensitive hygiene breeding program, and at the time it was kind of ahead of the time, and so unfortunately we didn't have genetic markers that we could continue that. But you as an industry tested that out, and you showed that 80% of the mites were not reproducing in the hives. So that's a huge jump forward from where we started, which was just less than 10%. We've looked at the products, we've looked at how to manage them and to get them into the country. We've looked at the how we use them. And to be honest, using them the way that the labels say is the most important thing that you can do. Half treatments will cause you problems. Reusing treatments will cause you problems. Using them the way that the label says is the most important thing. We've developed a little automated sticky ball counter. Of course, we don't really know exactly how that fits in with the rest of the industry, with the rest of the hive, so we're in the process of setting up a little bit of a program for that to understand the connection between that. And then probably the most important thing over all the time is this graph. This graph here and this graph here is the one that you need to look at. That's the one where you've got the distance between those two treatments. And if you slide that out and make that distance between the two treatments, your Varroa is going to grow too fast. So you need to make sure that that is shorter. Now, what that distance is in between now, we don't know, but it seems to be that we need to make sure that that treatment space is shorter. So today I'm going to be able to talk and answer to any of those questions, any of those things, previous research, the gap, the biology of the Varroa, and any other interesting thing that you'd like to talk about. Thank you.
Thanks, Michelle. You. Kia ora, I'm Phil Lester. I'm from Victoria University, Wellington, up here for some decent weather. I'm going to talk with you today about a couple of projects that are ongoing in our laboratory group. Both of these projects are going to be in Rose McGruddy's thesis, so thanks, Rose. It's awesome for your contributions and work, but also with a bunch of other people that are contributing from the lab group and from overseas. One of the first things I wanted to talk about and to get a little bit of a handle on or tell you about what's going on in the group is we get asked a lot, well, our treatments that aren't working quite so well. Our pesticides aren't going so well. what's going on? And so one of the questions that we've been looking at is around pesticide resistance in laboratory, right? Are increasing losses due to the efficacy of the products not working quite so well anymore? So this is one of the studies that we've been doing, and I'll quickly run through that, right? So this study had 3 components, 4 really. So the first component that we're looking at is, well, okay, are beekeepers satisfied with flumethrin and amitraz? We did see all those beekeepers as well experiencing increasing losses due to Varroa. We asked this of a group that's Pike has collected, so Pike was involved in the study as well. This is from the COLOS surveys, where we focused instead of on all beekeepers, just on commercial beekeepers, just on commercial ones, because we wanted to rule out the variability that's associated with hobbyists in there. And actually, the latest data from the commercial beekeepers showed that, well, are you satisfied with the products? 81% of commercial beekeepers indicated that they were satisfied or mostly satisfied with flumethrin, and 92% were satisfied with amitraz, or mostly satisfied with amitraz. So these products, according to the survey data that we're collecting, seem to be satisfying the vast majority of beekeepers. Are there known genetic mutations in the Varroa populations in New Zealand? So we did some genotyping work on the Varroa populations in New Zealand, and we can't find any of the known genetic mutations offering resistance to these mites that are seen overseas. Doesn't rule out there might be novel mutations in here in New Zealand. And the last component of what we've done here is looked at the toxicity of the products. And scientists do this a lot by calculating out an LC or an LD50, a lethal concentration or a lethal dose that would kill 50% of the populations. We compared the lethal concentrations that would kill 50% of the populations for the product now compared to some data that Michelle and Mark produced back in the early 2000s, soon after Varroa invaded the country. And what we saw there was for flumethrin, we're looking at, Mark and Michelle were finding about 12 micrograms per gram of product. We are finding estimates of about 156, so there seems to be some difference there, at least between those populations. And if you can compare that to this method was used in Italy in 1994, then they were finding 0.28 to 20. So there seems to be some difference in the toxicity of at least flumethrin from the Victoria University sites compared to what was seen in the early 2000s. What does that mean? It doesn't necessarily mean that the product is not working, the strips aren't working. It just means that the toxicity is different, and we need to be very careful about how we use it going on in the future. That's one of the studies I wanted to briefly touch on. The other is a study that I know a lot of people are really interested in terms of utilising the new products, double-stranded RNA, as a novel control method. There's a little video that's going on in there. Hopefully it's very small, but some of you will be able to see that. That's us putting the product in. We've done some trials now, some field trials of this product in New Zealand. What is double-stranded RNA? It's a very species-specific way of targeting Varroa, stopping protein production in Varroa mites. So we've utilised this. We've done a field trials and lots of lab trials and that sort of thing. We're pairing up with Greenlight Biosciences, who have got the patent on this in the US, and they've developed some pouches with sugar water and Varroa-specific double-stranded RNA that we're putting into the hives. The bees seem to like it and eat it up very quickly. We had 2 trial sites this year, one in Ashburton and one in Bulls. The one in Bulls is, as he says, pushing the wrong button. This one up here, so here we are at Bulls. This is on the x-axis, this is a week since spring, right, so going in, and then on the y-axis up there is the mite counts, it's really small, sorry. And the dotted line over here is sort of an economic threshold of about 5 mites per 300 bees. Right, so the yellow line is untreated hives, the blue line is a low dose of the double-strand RNA, the green is the high dose, the red is the Amatraz product, and there's that 500 mites. In bulls, wonderful bulls, it seemed to work reasonably well. We got up to about just a little bit above the economic threshold by the end of the season. So there's some product, some hope. In Ashburton, we were less happy with the results. We had really high mites in the control level, so you're looking at 60 mites per 300 bees. By the end of that, that did not spark joy as it was. We had somewhat lower levels in the high dose and even the Amatraz treatments. So I think the lessons that we've learnt here is that there's some hope. We have hope for this sort of product that's going on. In bulls, especially if you've got your mites under control, then you can potentially control. We still need to do work on that. But it's like anything, I think there's a dose response there. If you have lots of mites and lots of bees, big hives and lots of bees, then we're going to need to adjust the dosage of what we're going into it. So I think there's some potential for that sort of new product. This product is underway with under registration with the EPA in the US. We've got registration at the moment to allow us to do field trials here in New Zealand, but we're 2 or 3 years away from this product. But first, we'll need to demonstrate that it's effective and works and is safe and all that sort of thing.